is on multiple sequence alignments, how to construct them, and protein domains. So essentially, a multiple sequence alignment is the alignment of at least three or more sequences, either protein or nucleic acid. The important thing is that they should be homologs, that they need to have an evolutionary or share an evolutionary sort of common ancestor for any alignment to actually make sense. Otherwise, there's no point, because a multiple sequence alignment sort of shows you the changes that have occurred, if you like, within that sequence since they shared common ancestors. So, as the name suggests, you need to have a common ancestor. It actually allows us to find critical regions within those sequences as well, and kind of look for how well conserved certain areas are. So it tells us what changes occurred within the sequences since they last shared a common ancestor. They allow us to find important regions, critical residues, etc. And obviously, if residues are critical to function, then these tend to be more highly conserved within within those particular sequences. So that's actually really quite an important point. It's the basis, as we mentioned, for phylogenetic analysis, as you can see here, which I showed you last time, for constructing those trees. And they are used in protein structure prediction tools, actually in the background. So you won't necessarily know that that's what's happening, but in the background, when we use protein structural tools, they're actually using a multiple sequence alignment to similar sequences where we have solved structures and kind of trying to model those differences. So how do multiple sequence alignments work? Well, they essentially take all sequences, unlike a blast search, which we saw the other week, which is a pairwise alignment, so only using two sequences. Here we can align hundreds, if not thousands of sequences if we really wanted to. So it will take all those sequences and it will align them globally with each other or pairwise initially, and it will determine the most similar. So what it will do is it will optimally align the most two similar sequences together by trying them all out against each other, and it will then repeatedly add the next most similar sequence to the alignment, and it will just keep doing that over and over and over again until we have aligned all such sequences. So this is what the output from a multiple sequence alignment might look like. Importantly, on the left here, we have sequence identifiers. So in this case, it tends to be the species or kind of codes for the species. So we've got human at the top here, gorilla, all the way down to rabbit, mouse, rat, etc. Importantly, these aren't necessarily full homologs, but they at least have a domain, which I'll talk about later, that is actually homologous. The number identifiers for each amino acid residue, just note, by the way, that the amino acid residues are the single letter codes. And the numbers here denote the position, along with the numbers at the top here, of each amino acid. So here we start at amino acid number 1 from what we're looking at here, and go all the way to 57. In this case, these go to 58 and 59 because we've got a couple of gaps, which I'll talk about in a minute. Anyway, it doesn't matter. It's still numbered in that way. But importantly is actually this column here, because actually what this is telling us is that the human sequence, for example, we're starting at actually amino acid 100. So this amino acid number 1 is actually 100. And again, this 57 is actually 157 if we're looking at the full human protein. So when you give a number for an amino acid, you need to specify which sequence it's from. And if you are going to compare them to the human sequence, then you should always give the human sequence number as a reference. So in this case, 157. over here on the right. So that's just shown by this box here. You'll notice these arrows above. Don't worry too much about those for now. What you should worry about, though, are these gaps, which I alluded to earlier. And actually, what happens is, so in this case, you can see that leucine here, L, is present in this sequence, but actually it's missing in every other sequence. So in order to allow the rest of the sequences, both before that leucine and after, to be optimally aligned, you have to insert gaps sort of in these sequences to allow that alignment to happen. And those gaps are actually taken into account when we look at the scoring matrix later on. So that's all you need to worry about too much, but that's why the numbers here are slightly skewed here, because there are actually gaps at various points. And you can see also that this alignment runs actually into a separate block down here. So again, this sort of ends at 57, 58, 59, 60 here, and it continues to 117 in the case of the human. But importantly, not all amino acid changes, when we see a change, are actually equal. So if we have something such as aspartate, so it's a fairly rudimentary side chain, it's negatively charged, however. If we were to swap this out for something such as a glutamate, actually it's a very, very similar structure. It only has one more sort of carbon group. It's still negatively charged. It's still pretty much the same size. It's not going to have a drastic effect at all. So you need to switch your brains on when you look at amino acid changes. And just because there's a different amino acid there doesn't mean that it's going to have a massive impact on the function. If, however, we were to swap to something such as a phenylalanine, you can see this phenylalanine is actually, instead of being negatively or charged, it's actually neutral. It's quite large in comparison. It's aromatic. It's got this benzyl group with these kind of unsaturated bonds, and it's hydrophobic. So actually, this is very different to the aspartate there. So we can say that this particular change to phenylalanine is most likely to have an impact on protein function, if indeed this particular amino acid, this particular aspartate, is found within a region of the protein that actually has a real function, and it's not just kind of a linker region, which we'll talk about later on. So just to remind you, this table might come in useful during the computer practical. It gives you the full name, the 3-letter code, and the 1-letter code. So arginine, Arg, and R are all interchangeable. Generally speaking, don't forget it's the single letter code that we use. And it will give you information about them. So again, charged side chains, we've got the positive and the negatives. We've got polar uncharged chains, so things with OH groups and NH2 groups, etc. And then we've got sort of hydrophobic side chains which don't like water at all. And again, our phenylalanine comes in there. So I would keep this table sort of close to hand when you're doing the practical. Special cases are here for various reasons. I'm not going to talk too much about them. One that comes up, however, is cysteine, which is important because of the sort of thiol group down here, which allows formation of a disulfide bond with another cysteine residue if they are close enough together. Turns out we can actually quantify how big or not these differences are by using a scoring matrix. So Blossom is a very common one, which I'll show you now. And all this is, it gives a number. So the amino acid residues here are the single letter codes. So if we were to take something like glutamic acid, so this E here, if we were to change it to something like an aspartic acid, D, it's given a sort of 2, it's a sort of positive score. It's a sort of decent change, but actually not drastic. If we were to change to something such as a phenylalanine down here, which is very different, it actually gets a negative score, in which case. Importantly, cysteine, for example, here, if we don't change cysteine, it actually gets a very positive score. Now, that's because it's so positive, because actually cysteine is incredibly well conserved within proteins. It's very important because of its ability to form disulfide bridges. And you can see here that when we eventually get an alignment, we can color code it according to this particular matrix called Blossom, and it will tell you what amino acids are similar in the case of the green and the blue colours, or in the case of the white, here we've got glutamic acid and leucine, so EE for glutamic acid and leucine. These are very different, hence why these are white and these are blue. So you can colour code it for all sorts of reasons, but that's a good example and will be quite useful to you. So revise your amino acid chemistry and get familiar with these one-letter abbreviations for amino acids. Just to point out, when you're looking at amino acid and you want to look at where the side chain is, if it's not just the side chain that's shown, you should always look for the NH3 plus group and the COO minus group. Don't forget, amino acids at physiological pH are zwitterions. They have both of these charges. We should look for the hydrogen, and then the other group, everything off that other group is the R group. So in this case, it's CH2, and then you have this kind of ring structure, this kind of structure here. So this is histidine, etc. So again, proline here, COO minus rather, NH2. So the R group here starts here and actually links back to the NH3 plus group here. So that's just showing that there. So again, let's just look at two amino acids just to sort of labour the point. Here we've got glycine. It's the most simple amino acid because its side chain is simply a hydrogen atom. Over here we've got something called proline, which is rather unique in its ability to introduce bends into proteins, but actually its R group is very different, which is all I'm trying to kind of point out here. So again, NH2COO minus, the R group actually starts at this CH2 and it connects all the way through to the NH group here. So it's actually quite a significant R group in comparison to a single hydrogen atom. And that can be seen here when we look at the structures. You can see the single hydrogen atom here, and actually the whole sort of looped sort of ring structure here is quite bulky, it's quite big. You can imagine it's quite limited in the way that it can move. So how do you make a multiple sequence alignment? Well, there's lots of different programs. We're going to use something called MAFT, which stands for Multiple Alignment Using Fast Fourier Transform, rolls off the tongue. And the typical workflow would look like this. You would essentially, first of all, find homologs. We've done that using BLAST. Once you've got homologs or potential homologs, you make a set of those sequences on a text viewer. You then align them using our program, in this case MAFT, and you analyze the results looking at them in a nice little table like this. Just a note, this is quite important, so I'll note it down. This particular view here we don't get from using MAFT. MAFT will simply align your sequences. Actually, this view here, you need to input the output from MAFT into a specialized viewer, something called JALView, which we'll look at. Download that before next session, and that will allow you to actually look at it like this, which is much more useful. So just a quick one on MAFT. It will look like this when you go to the website. Make sure you click the online version up here. Don't download it, there's no need to. Once you've clicked the online version here, you put your input sequences in here. So once you've got all of your sequences you wish to align, pop them in here, and then you just click go, and it will align them for you. You can change the details down here, but we're not going to worry about it. So the final thing I sort of want to talk about actually is what a protein domain is, which we'll come across in the practical as well. It's an extension to your kind of knowledge of protein structure, but you do need to be aware of it for the computer practical. And essentially it's defined as this. It's a part of a polypeptide chain that can independently fold into a stable, compact tertiary structure, often with its own, and this is important, often with its own specific function. So what we mean by independently folding is if we were to chop it up and chop that domain out of the rest of the protein, it would still, under the right conditions, fold into the same shape. Importantly, this is one example called as part of a zinc finger domain, which actually its function is actually very good actually at binding into the major groove of DNA, so it can bind to DNA. And as you can see here, there are loads of different functions. This is not an exhaustive list. So we've got DNA binding there. Things with dead boxes are able to bind to RNA. Walker A and Walker B motifs, or sort of domains, are able to bind and hydrolyze ATP. So any enzyme that requires ATP hydrolysis, so things like the ribosome, various things like that, that actually need to hydrolyze ATP in order to function or translocate or things like that, will have Walker A and Walker B motifs or kind of domains within them. Metal ion binding and helicase activity as well. So RNA polymerase, for example, doesn't recruit a separate helicase before it can actually access the bases within the duplex to catalyze transcription. To catalyze transcription and creation of RNA. It actually, within its actual structure, it has a domain that has helicase activity. So we often say it has intrinsic helicase activity. So this is an example. This is DNA polymerase from E. coli here. And you can see it's split into two domains, actually. The domain up here is the polymerization domain. It kind of looks like a hand, right? It's sort of got fingers, a palm, and a thumb. And this domain allows the normal function of DNA polymerase, which is to catalyze the formation of phosphodiester bonds between sort of nucleotides. But the exonuclease domain down here is a separate domain. It's still part of the same protein. It just has it folds into its own little shape and has its own function. That's what a domain is. If we were to cut this in half, both of these domains would still fold into their relevant shapes. And it's an exonuclease, so it will degrade DNA from the end. And what this is useful or used for is if the polymerase domain makes a mistake, it puts the wrong base in during replication, it can actually flick the strand that it's elongating into this exonuclease domain. It kind of pushes it into here, and this exonuclease domain can sort of chew back that base, the incorrect base that was put in. It can then flip the strand back into the polymerization pocket domain, and then the polymerase can have another go at putting in the right base. Something called proofreading, which is really quite cool. So my fairly rubbish example, but hopefully you get the point, is that if this lady here that's holding this Coke bottle is a protein, and her job is to unscrew and open the can of or the bottle of Coca-Cola, she has two domains within her kind of overall structure. This domain at the top here actually can grab hold of the cap, and it's specific for the cap, and it can turn to allow the movement of the cap. But the left hand here acts as the second domain, if you like, and actually its job is to bind the bottle in the first place and keep it steady to allow domain 1 to do its function. So this is just one sort of example that you might want to be aware of. It just shows kind of domain organization quite nicely, which is the CD4 protein, which is found on a T lymphocyte. And it's quite interesting, actually. It sort of specifies the immune response to be a helper T cell response rather than a cytotoxic T cell response, which is really interesting. But anyway, you can see here when you look at this particular protein, they look like beads on a string, right? There's 4 domains. They're separated by what we call linkers, which are just kind of strands of amino acids that actually don't really conform to any domain. They just allow sort of connections between domains. And you can see that the 4 domains here I've coloured in kind of different colours, just so you can see them, which is quite nice. So, how do we find protein domains? Well, we use a similar search to a BLAST search, where you can search by text or similarity. Generally, we'll search by similarity. But the domain database we use is something called the Protein Families Database, or PFAM, and it actually looks like this. And what you do is you would click sequence search up here, and that would bring up don't worry about keyword search, bring up a box like this, and you simply paste a protein sequence into here, single letter amino acid code. You click go, and it will sort of use e-values and that kind of thing. Don't worry too much about that, but it will then ping back what the domains look like. This is one such example. I put in something called GCN4, which is a transcriptional activator protein from yeast. And you can see it gives you a kind of rough organization here. You can click on these boxes and it will tell you what they are. More detail on that in a minute. But the cartoon of it down here shows that there are actually 2 domains, and you can see those domains in a moment. One domain is a DNA binding domain, and the other is transcriptional activation domain. So the DNA binding domain will bind to DNA, and the transcriptional activation domain will interact with another monomer, and it will allow recruitment of other proteins to modify chromatin structure to therefore allow the DNA to be more accessible for RNA polymerase. So this is just a kind of zoomed in version, and I've coloured in purple here the DNA binding domain. Each of these are monomers, so this is actually a dimer, so this is two lots of sort of tertiary structure coming together to function via a leucine zipper region here. So these side chains here in blue and yellow are leucines, and they kind of zip together to allow them to function. But this is the DNA binding domain in purple. You can see it's binding to the major groove of DNA. And up here in grey is the transcriptional activation domain, which will then interact with other proteins to recruit them to the DNA duplex at this particular region. And it will then eventually allow DNA to become more accessible for RNA polymerase. So what does the output look like? It looks like this. So you get this cartoon, as I mentioned earlier. And in this case, I'm looking for a HEL 308 helicase in an archaeon that I work on in the lab. And it has a dead box domain, which actually allows binding to RNA. So it's actually a sort of, it can kind of unwind RNA that binds to DNA. And there's the helicase domain as well. So that's the domain that will allow it to form a sort of hexameric ring, which is typical of a eukaryotic helicase, but actually it occurs in archaea as well, which are prokaryotic. And you can see the other domain here, which we're not going to worry too much about, but it's a helix, hairpin helix domain. I'm just going to use this this is another example of a Argonaut protein. Don't worry too much about that, but it's got a PAS domain and a PIWI domain, so two domains within this protein. And actually, you can look at the family here, so you can click on these two links, and it will give you a page, which I'll show you in a minute as to what they do and give you information. Don't worry about the rest of it. What I'd worry about the alignment here, so it will tell you where these particular domains start and end within the sequence. So you can see it's the PAS domain first, followed by the PIWI domain, and exactly what amino acids they start and stop at. Hidden Markov models you don't need to worry about, but just be aware you're given a score similar to a blast search, and importantly an e-value. So I would just worry about those particular regions. I'd worry about being able to highlight these and click on these to kind of get information. Be sort of concerned about where the alignment starts and ends for each domain within the protein, and the e-value as well. So again, we want that to be a low value. If you were to click on this particular PAS link here, or indeed click on that, you would get to a page like this where it gives you much information. There's kind of links at the side and the top where you can sort of look for interactions between other proteins, what species they occur in, and also importantly for you, perhaps later down the line, is it will give you a link to a protein databank entry where you can get the sort of solved crystal structure for this protein, and you can model it in our protein modeling session later on. So that's just a bit of useful stuff for you. So this is the summary. This is sort of what I've talked about. Multiple sequence alignments and what they are, how to do them. I've briefly introduced a scoring matrix to you and protein domains, as well as looking at PFAM in terms of how you look for those domains within proteins. Before next week, please do, if you haven't already, download JALView. Links are on the team site and in the handbook for the course. And then when you do the practical, again, just open up the practical. It's a set of instructions with an associated kind of quiz as you go through. So follow the instructions and complete the quiz, and I will be on hand to give you assistance either in the session or email me if not, and we can sort out another time.